The cell cycle can be thought of as the life cycle of a cell. It is made up of two main parts: interphase, where the cell prepares for cell division, and M phase, where the division process actually occurs. Within interphase, there are four main stages. The cell can either enter G0, where it exits the cycle and stops proliferating, or it can enter G1, where the cell prepares for DNA replication. Then the cell enters S phase, which is the DNA replication phase, and progresses to G2, where the cell prepares for mitosis. There are four main checkpoints that occur throughout the cell cycle: at the G1S boundary, in the S phase, and during the G2M phases. The progression of the cell through the cell cycle is regulated by two important proteins, cyclin-dependent kinases or CDKs and cyclins. The different CDK cyclin complexes either activate or inactivate different enzymes by adding a phosphate group to the enzyme. Various faults can occur in different parts of the cell cycle, but mostly they affect the G1 phase. P53 is an important transcription factor responsible for halting cell cycle proliferation when DNA damage occurs. To do this, it encodes the P21 protein, which binds to CDK and inhibits its activity, which causes the cell cycle to halt until DNA damage is repaired or cell death is induced. A missense mutation in the P53 gene results in P53 proteins unable to bind to DNA. When this occurs, P53 can no longer induce DNA repair and apoptosis, and therefore the cell will progress through the cell cycle with damaged or mutated DNA, which is one of the main causes of cancer. Another major fault that occurs in the cell cycle is a mutation of the retinoblastoma protein. Retinoblastoma protein is a tumor suppressor protein whose primary function is to prevent excessive cell growth by inhibiting the cell cycle progression until a cell is ready to divide. To do this, RB binds to and inhibits a transcription factor called E2FDP, preventing the cell from being pushed into S phase and keeping it in the G1 phase, and then hyperphosphorylating, allowing E2FDP to become active and push the cell through the cell cycle. In cancerous cells, there is a mutation of the RB1 gene found on chromosome 13, which can affect the E2F transcription factor, allowing for altered cell cycle progression and subsequent cell growth. MYC is a proto-oncogene that encodes nuclear phosphoprotein that plays a role in cell cycle progression, apoptosis, and cellular transformation. In partnership with MAX, it activates transcription by increasing the expression of cyclin D2 and CDK4. It also increases the expression of E2F1, E2F2, and E2F3. Application of the MYC gene is frequently observed in numerous human cancers and results in increased expression of cyclin D2, CDK4, and E2F1, 2, and 3, which leads to continuous cell cycle progression. Translocations involving this gene are associated with Burkitt lymphoma and multiple myeloma in human patients.